So we are starting chapter 7. It's going to be our biochemistry chapter. Most of this should be review for you. So it does gloss over some concepts that I hope you have gotten previously in an earlier biology class or an earlier chemistry class, because we are talking about stuff that you hopefully have heard about, and where this should be a refresher for you. So we will get started on the first one. And it is, where did life come from? How do we have these macromolecules that we're going to be talking about in this chapter? And we had some different theories. One theory is that life on Earth, that we know is 4.6 billion years old, by the way, that life on Earth may have originally started outside of the planet, and that we had life come from outside, and those basic molecules came from outer space, landed on Earth, and those were our building blocks. There is another theory that life began on Earth and it has always been here, and that's how it developed. And so that theory is reinforced by this slide, this first presentation slide here, with the Miller-Urey experiment. And so what Miller-Urey did is they tried to recreate that original Earth atmosphere. So 4.6 billion years ago, the Earth did not look like it did. It did not have the atmosphere like we have. The oxygen levels were low. They were not like they are now. None of this looked like this. So it was a boiling rock with noxious gas out there, and it was pretty hostile. So Miller-Urey set about to recreate these conditions in a scientific form, and so he has um, what he tried to recreate what he believes was in the seawater at the time. So he recreated a primitive ocean, and that there were lightning storms going on at the time. So he has electricity involved in this experiment, and he heated up the ocean water, because there was, volcanoes happening and they were spewing noxious chemicals out there. So we're recreating all of these events: volcanoes, poisonous gas, ocean water, and lightning, and we're putting it all in this experiment for Miller-Urey. And they heat up the ocean water, and we're going to set it to steaming and put that ocean water vapor up into the atmosphere. And then we would try and simulate the atmosphere. Atmosphere comes over this with this ocean gas up in the air, and then sparks from, say, pretend lightning interact with that water, and then it's going to condense, it's going to cool, and it's going to condense, and then we're going to end up with water down here after we have boiled it, simulated in a primitive atmosphere, electrocuted it, and then condensed it. What did the water at the end look like? And the water at the end contained organic compounds. It contained some of the building blocks, some of the amino acids that we use as building blocks for proteins. We were able to recreate, at least Miller-Urey was able to recreate in this experiment. So this was trying to prove that the Earth's atmosphere, that we could recreate the Earth's atmosphere and get these primitive building blocks, that life could have started here all along, with the Miller-Urey experiment. So let's talk about the building blocks and all those different macromolecules. So when we talk about molecules, we got to go back to the building blocks of a molecule, and those are elements. Those things are going to be matter that cannot be broken down into simpler substances. So now I'm just reading to you off of there. Hopefully you know about these things, that you've learned about them in biology or chemistry before. So these are the basics. Elements are the basics, and we do have subatomic particles, protons, neutrons, and electrons. Every element is different because every element is defined by the number of protons that that element has. If you have 2 protons or 3 protons, you are 2 different elements. And every element is going to have a chemical symbol on that periodic table. Some of them are in English and it makes sense, O for oxygen, but others are from previous languages, and it may not make sense, like natrium for sodium. Instead of having an S, S is sulfur, sodium is Na, so it might be coming from another language. Again, this is all refresher stuff, hopefully, for you. You've heard about this in high school. You know what I'm talking about. Out of the elements, we are going to have some elements in our body that we use more than others. The ones that we use more of are going to be called principal elements. Elements that we still need, that are critical to us, but we don't need as much, are going to be called trace elements. So we've got a whole table over here of principal elements, and we can see there are these right here: oxygen, carbon, hydrogen, and nitrogen. These are the top four elements in organic compounds, the top four things that every living thing has got these things in them. So on a test question, if I was going to ask you which ones are more important, principal or trace, we need more of principal than we need of trace. They're all important, but we need more of the principal ones than the trace elements. And which specific ones are the most common? There are four: oxygen, carbon, hydrogen, nitrogen. So know those four for the test. CHON. How do we get molecules? So molecules are going to be when two elements come together, and depending on their valence shell, how many electrons they have in their valence shell, or how reactive they're going to be. Do they have places for one opening? Are there two openings? Do we have room for multiple elements to bond to another element? So carbon is a pretty popular element because it has four places that we can bond to. It may be a single bond or a double bond. So we've got one bond here and one bond here with this oxygen. So we double bonded. And so that takes up two places, one here and one here. And then we've got another spot here and another spot here where we've got another double bond. So we've got four bonds going on, two double bonds for this oxygen and another two double bonds for this oxygen to take up all the places on this valence shell. Nitrogen, we've got 1, 2, 3 spots here that we bonded with, and you can see 1, 2, 3 if we drew them like there. And then oxygen, a molecule of oxygen has 2, 1, 2, and double bonds between them. When they are bonding, there are ionic bonds, and there are hydrogen bonds, and there are covalent bonds. When we talk about organic molecules, all those guys are going to use covalent bonds. And if you remember from previous classes, covalent bonds involve sharing the electron back and forth. It may not be an even sharing, it may be polar, where one element hogs it on their side more than the other, like water, but they're still going to share. So organic molecules are going to have carbon. All organic molecules will have carbon, but they also are going to be bound to a hydrogen somewhere, somehow, somewhere on that molecule, and that's what makes it organic. So we're going to have a carbon backbone. There will be carbon somewhere on that molecule. We'll also have a hydrogen, and these are going to be bound by covalent bonds where they share the electrons back and forth. That's what's going to make it an organic molecule. Inorganic molecules may have carbon, because you can't just make the blanket statement. Every time you make a blanket statement, there's always an exception. So if you just said, oh, carbon on a backbone, and here we've got CO2, that's got to be organic because it's got carbon in it. No, it doesn't have the hydrogen. So CO2, although it does have carbon, is an inorganic molecule. This guy is in this category. But all of our other guys with carbon backbones bound to a hydrogen somewhere, those are going to be organic, and they're sharing their electrons back and forth in covalent bonds. Isomers. So if we talked about glucose, if we wrote this down, would be C6H12O6. For our carbs are always going to be in a 1 to 2 to 1 ratio. If we took C6H12O6 and we made glucose, this is how it would be if we took it out of its ring formation and drew it as a chain. This is what glucose looks like. But this is galactose, and this is fructose, and they all still have this chemical formula C6H12O6, but you can see they are built differently. These are isomers, so they have the same building blocks, but you put them together differently. Those are isomers. So glucose is the same as fructose, is the same as galactose, but we build them differently even though they have the same building blocks. So these are structural isomers, same building blocks, but we built them differently. And then there are stereoisomers, enantiomers, and these guys are isomers, but they are going to be a mirror image. So just like it's says in this picture right here, they are like a reflection of the other side. So what they do is they look like they're rotating on a planet, on the planet of light. We've got the D and the L, and the textbook goes into tell you the backstory on the D and the L. But why we would want to talk about isomers are that some of them, one isomer, one type of it, like the D-glucose versus an L-glucose, D-glucose may have different properties, different structural properties versus the L-glucose. Certain types of bacteria can only take care of, can only metabolize maybe one of the isomers and not both of them. Maybe they can only pick up L-glucose instead of D-glucose. So isomers, they're mirror images of them, but sometimes only one of the isomers would work and not both of them. Functional groups. These are things that will attach onto um, molecules that attach onto your chemical formula, and they are made out of certain things and they are named after their structural components, these functional groups. So there are a whole bunch of them in here. If you took chemistry at Georgia Highlands, you would need to memorize these things. I'm not going to make you memorize all of these things, but it's nice to know what some of these are, like amino. If you see nitrogen, I want you to think of amino groups. We talked about ester and ether. Remember when we were talking about bacteria versus archaea, those ester and ethers. We might talk about methyl if we had methyls or methanes. But those are the different functional groups. I'm not going to make you memorize these things. Hopefully you've already seen them before. How do we make macromolecules? That's the rest of this chapter is the macromolecules. So macromolecules are going to be built off of smaller molecules, of course, because macro means big. So we are going to take two things and put them together. And when we take these two things, this is a monomer and this is a monomer, and we're going to put them together. If we take one glucose and this other glucose, and we want to bring them together, we can see we've got this and this. So what if we pulled that out? If we pulled that water out, we have pulled the water out, we have dehydrated it. So if you are thirsty, you are lacking water. If we have pulled the water out, we have dehydrated this. And when we dehydrated it and we pulled the water out, we synthesized another different molecule. So we had, this was glucose, here's glucose, and another molecule who is separate of glucose. And we want to bring them together to build a bigger, larger molecule. We want to take an individual monosaccharide and another monosaccharide, and we want to make a disaccharide. We're going to pull out the water, dehydration, and when we dehydrate it, we synthesize a bigger, larger molecule, a disaccharide, and that disaccharide is called maltose. So if you see a reaction that pulls water out of something to make something bigger, to make a larger, bigger molecule, you are seeing dehydration synthesis. And all of our macromolecules are going to be like this. One molecule plus a second, one molecule plus a second molecule is going to give us this bigger molecule, disaccharide. And then maybe we tack on more and we tack on more, and eventually we have hundreds of these put together. Then we end up with polysaccharides. So just to refresh, dehydration synthesis is pulling water out to bond two molecules together. Different types of macros. We're going to talk about carbs in this video, and then the next video we'll go into lipids, our fats, nucleic acids, proteins, and how we identify these things. But these are the 4 macros. If you are maybe watching what you eat, watching your macros, what are you in, how much carbs are you bringing into your body, how many fat grams are you taking in on a daily basis, how much protein are you taking. These are the macros. All right, so our case study leading off for this chapter is Penny. She is 16. She went to her doctor about an itchy rash, and she does have a history of allergies. So the doctor saw that she had suntan lines and asked if she used a different sunscreen, something out of her norm. And she said that she did use a different sunscreen. So the doctor said that she's just allergic to her sunscreen. She's got eczema going on. And so he said, stop using that sunscreen, and gave her an over the counter cream to help with the itching. So we'll check and see at Penny and how her rash is doing, and hopefully it will get better, but we're going to keep going. All right, the next section is our first macromolecule, and that is carbs. And these are going to be the most abundant molecule on that we are going to use, biomolecule. Um, Carbs are your friend. How do we describe carbs? So we are going to talk about carbs, and we can count up how many carbons there are in the carbohydrates. So this one has 1, 2, 3. This one also has 1, 2, 3. This one has 1, 2, 3. So if you got 3 carbons, you're try something because you have 3 carbons on your backbone. And then where is the carbonyl group? Here, your aldose. Here, it's a ketose. And if we built this guy over here, glyceraldehyde, versus this guy over here, I'm gonna slaughter that one. Dihydroxyacetone. I got it. It's just where is that put? So three carbons. Where's that carbonyl group? Names it something different. Maybe we have not 3 carbons. Let's see how many we have here. 1, 2, 3, 4, 5. So we have 5 carbons. So this is a pentose. Once you get 5, 6 carbons, you'll see them written as a chain, or you may also see them written in ring formation, like we had over here. These are sugars, but they're written in ring formation. If you see them in the ring formation, you assume that there is a carbon every time you get to one of those bins. You're assuming that they are there somewhere, that there's a carbon at this bin here or this bin here. So we could have 3 triose, we could have 5 as a pentose, we could have 6 because this is glucose. And so 6 would be a hexose. So how many carbon do we have as the backbone for monosaccharides? And mono means one. It is one. Sweet is literally what it means. So these are simple sugars. These are building blocks for carbs. We can see we're talking about how it was a chain formation, linear, or you can put it in as a ring or cyclic structure over here. Moving on, if we put 1 and 1 together, we make 2. So here's our disaccharides. This is the example that they gave us beforehand. We have 1 molecule of glucose and a second molecule of glucose. We put them together because we pulled out the water, and now we did dehydration synthesis. When we dehydrate the 2 molecules of glucose and pull out the molecule of water, we end up with this entire molecule, and that is called maltose. And we can do the same thing to create other sugars, other disaccharides. So we have lactose over here, we have sucrose over here. Lactose is your milk sugar if you are lacking the enzyme that breaks down milk sugars. You are lactose intolerant. If you do not have lactase, in your body, you don't have that enzyme to break it down, so you're lactose intolerant. And what ends up happening is the bacteria in your gut break it down for you, and that can cause digestive upset when you rely on the bacteria to break it down instead of the enzyme lactase to break it down for you. So these are disaccharides, 2 sugars for disaccharides. Moving on up to where we got a couple hundred of these things, and these are polysaccharides. Every time these things are bound, when we talk about that dehydration synthesis, if we're talking about dehydration synthesis for sugars, these are called glycosidic bonds. So they are still dehydration synthesis because we're pulling out the water and making a bigger molecule, and that's dehydration synthesis. But if we're talking specifically about two sugars that are bound together, the bond between the two sugars that are bound together are glycosidic bonds. So we've done a ton of glycosidic bonds. We're on polysaccharides, and these things have hundreds of sugar molecules all bound together. So there's different types of polysaccharides out there. These are the three common ones that I want y'all to know. So our carbs are going to be a form of energy storage, short-term energy storage. So if we ate a big meal and your blood sugar is really high because you just digested that meal and you've broken down all those complex carbs into simple sugars, your body's going to use those simple sugars. And what doesn't get used is going to get stored away. As if we're animals or if we are bacteria, we store our sugar away as glycogen. That's our animal sugar, animal bacteria sugar storage molecule. If we were a plant, we would store our sugar away as starch. So if you think about starch, one of the things that pops into my head is potatoes and french fries. They're heavy, starchy components in those potatoes. So starch is the plant's version of our glycogen. It's their polysaccharide to store away their energy for short-term energy storage. So short-term energy storage for animals, glycogen. Short-term energy storage for starch, for plants is starch. And then there is also a polysaccharide that we've mentioned a few times, and we said that cellulose is going to be a polysaccharide that plants use on their cell walls. And we said that fungi use a different type of polysaccharide that's not here in these PowerPoints. It's called chitin, and that's on fungi cell walls. Plants are going to use cellulose on their cell walls. But as far as energy storage, plants use starch for energy storage. Animals and bacteria are going to use glycogen for energy storage. So that covers this video. We'll come back and do lipids, proteins, and testing for it in the